Okay, here's the last portion of the virus PowerPoint. And the rest of this is going to be primarily eukaryotic. So I've already taken you through bacterial viruses and how they take over host cells. So now we're going to move over to non-bacterial cells and their viruses. So here's a look at stages of a lytic replication cycle. Remember, in bacteria there's both a lytic and a lysogenic replication cycle. In eukaryotic cells, there is no lysogenic, it's just lytic. Although the cell, it does not necessarily have to die right away, as do bacteria. Alright, so remember, let me take you back through the bacteriophages. Attachment, entry of nucleic acid, synthesis of viral proteins and nucleic acid, assembly of those proteins, and then release from the host cell. So you'll notice that there's something different here, and that is uncoating. So let me take you through this little variation of a cycle here. Here is the beginning. Attachment. Notice that the entire virus, it may have an envelope, it may not, but the whole virus, including its capsid, goes into the cell. That's called entry. Once inside the cell, the viral structure comes out of the vacuole, or the little vesicle that it is inside of. That is called uncoating. Capsid is stripped off, freeing the nucleic acid of the virus, which you see here as purple strands. Those purple strands, the viral genome, the genes are red, viral proteins made. You see those right here. Viral genetic information made. You see that as purple right here. They are then packaged together, making new viruses. That's called assembly right there. And then, of course, there has to be release. So notice that the virus is budding off of the host cell, taking some of the host cell membrane with it. Here is another viral life cycle. Here is our virus right there. And here is the attachment. You can see the receptors on the outside of the membrane right there. The virus is taken in. Notice that the host cell is actually bringing the virus in by endocytosis, forming a vesicle around it. Then, step 3, vesicle removed, capsid broken open, viral nucleic acid released. It is now going to be red, making viral proteins, these guys right here, and viral nucleic acid, in this case RNA, assembly, and budding. There's your release right there. Okay, so attachment. Over here on the right you see a virus. Little receptors, looks like little black triangle things on the outside. And notice the receptors of the host cell, blue structures. That allows the entry of the virus. Viruses have very specific host cells. That specificity is due to the receptors on the host cells. So, for example, on the left over here you see a T helper cell. And T helper cells of the immune system have a special receptor called CD4s on the outside of their membrane. That receptor is where HIV attaches. HIV is very, very specific for its host cells. T helper cells are its main host cell. So looking down the list here, you can see a whole variety of viruses and receptor structures that they bind to on host cells. These receptors have different functions in the host cells. They may be attachment points for chemicals. They may be attachment points for antibodies. They may be where neurotransmitters attach. All sorts of different receptors. They weren't made for viruses to attach. Okay, so here is now entry and encoding. There are three mechanisms here. One is for the naked, non-envelope viruses. That's up at the top. You can see that it looks very much like the way bacteriophages got into host cells. The virus attaches to its host cell. Then the nucleic acid appears to go through the capsid wall. And through the cell membrane and ends up inside of the cell. That's called penetration. Now, for the envelope viruses, which you see here, and you see here, there are two major mechanisms. Let's go membrane fusion. The capsid is inside. Look at the capsid right there with the nucleic acid. Now remember, the envelope is mainly lipid. And what is a cell membrane? Mainly lipid. Alright, so the lipid envelope attaches to the lipid membrane. You see that happening right there. They fuse together. That opens up a door for the viral structure now to waltz into the host cell. And there's your uncoating. That's membrane fusion. Now, down at the bottom, you see the mechanism that you've seen before on pictures, which is endocytosis. Here the virus is actually brought into the host cell. There it is being brought in. A vesicle forms around it, so there's an outer vesicle, and then there is the actual envelope of the virus right there. They fuse together because, remember, membrane, lipid, even vesicles inside the cell are lipid, the walls of them. And cell membrane, that is, the envelope is lipid and the cell membrane is lipid. So here you see the two fuse right there, forming one envelope of virus, vesicle membrane wall of vesicle, and again, there's your capsid and virus structure inside the cell. Okay, so synthesis of the new viruses, proteins. Lots of different strategies. Not going to get into all of them. I'm going to take you through some of them in lecture. But it depends, the strategy that the virus takes in the takeover of the host cell depends on whether you're talking RNA or DNA virus, and whether it's single-stranded or double-stranded RNA or DNA. Are there any viral enzymes that help with the host cell takeover? And there are some unique viruses that we'll talk about that have some very different kinds of actions that make them unique among even the viruses. Alright, so we're going to start over here. Here is your virus. This happens to be a single-stranded RNA virus. It's a poliovirus. The RNA is in gold. So, this is penetration, direct penetration. The nucleic acid comes in through the membrane. You see it now inside the cell. This is called plus RNA. In other words, plus RNA is identical to mRNA. In fact, it acts as mRNA. So here is the viral genome that came in. Again, it's a plus strand, meaning mRNA. This mRNA now can be read by ribosomes, right here, for the production of viral proteins. In addition, this mRNA now is going to be the template to make minus RNA, which is the complementary strand of RNA. So again, if you have an mRNA molecule right here, And let's say, just to make things easy, it is A, all the way down the strand. Then the complementary strand of the minus RNA would be U, whatever. Okay, now, so that's what we've got right here. Now this strand, remember, is not viral genomic information. It's the complementary strand. So this now is used to make the complementary strand. We're back now to A. This is viral genomic information. And we can make 30, 50, 100 copies. However many viral baby structures you want to make. So now we've got the proteins being made up here, viral genome made here. Here is the assembly right there. Here is our new virus being made. And then, of course, exit, release. Okay, here is another strategy. This happens to be something more like a herpes virus, as a matter of fact. And look down here. Here is double-stranded DNA of the herpes virus. So look right here. By the way, this is cytoplasm that you're looking at out here. All right, so this is happening inside of the nucleus. Most of it's happening in the nucleus. Because remember, this is where DNA is. So double-stranded DNA goes through a nuclear pore. There it is right there. That's the viral genome material. Notice that it opens up just like your DNA would, and one of the strands, the sense strand, is used to make an mRNA strand. That mRNA strand goes out, just like it would in your own cells, uses the host cell's ribosomes, viral proteins constructed. Okay. Now, what happens to that double-stranded DNA? The other thing that's got to happen is we've got to make more double-stranded DNA to be packaged in the virus. So, just like in your cells, the two strands come apart. Here's strand 1, here's strand 2. Each of these strands now makes a complementary strand in black. We now have 2 whole copies of viral genetic information. These 2 can separate. Now we have 4 single strands, and those 4 single strands make complementary strands. Now we have 4 copies of genomic DNA, and then we can have 8, and then we can have 16, and so on. Again, however many viral structures are coded for. So here are all the components we need, the proteins, the capsid proteins, and the DNA, and there's assembly, and then of course virus goes out of the cell. Okay, so I've taken you through a couple of the mechanisms, and I'll take you through a couple more in lecture. All right, so here are the major groups of the animal viruses. Again, they're broken down into groups basically by RNA versus DNA, single strand, double strand, and then some kind of unique mechanisms that they might have. Here is the HIV representative. It is a reverse transcriptase virus. It's an RNA virus, and it does something odd. It directs the host cell to make DNA from RNA. That's why it's called reverse transcription. So in this scenario, here is your virus right there. The genome is in gold and black inside of the viral structure. Here it is right there, single strand RNA. It now is going to be used as the template to make a DNA. Once it has one strand of DNA, that DNA strand is used to make the complementary DNA strand. So now we have double-stranded DNA. This double-stranded DNA now is going to go into the cell nucleus, where it can integrate into the host cell chromosome, and it can stay there not doing anything, dormant, for a long time, or at any point in time it can be read, single-strand RNA can be made from that DNA, just like mRNA would be made. It codes for new baby viruses, proteins, more single-strand RNA is made. Here is our assembly that's going on right there of the virus. Here's budding out from the host cell membrane. And when it buds out, it carries some of the proteins on the outside of the membrane that it's directed the host cell to make, to put on its own membrane. And there's our virus right there. Okay, so now we're at assembly and release. Here's a really good view of the assembly and release of viruses. You're looking at an infected cell. The little viruses are the small opaque structures. There must be 150 at least inside that cell. You can see it's ruptured open. Many times in assembly, there will be some unique structures inside the cell that can be seen that designate a viral infection. They're called inclusions, viral inclusions. You can see right over here, this is the CMV virus. It's in the herpes virus group, cytomegalovirus. Here is our infected cell, and it's got some little inclusions. Again, these are viruses being assembled inside the cell. So the infected cell looks very different than normal. Down here at the bottom is a neuron. That's this big guy right here. And there's the nucleus. But look at this guy right over here. This is a bunch of aggregated, assembled viruses, or viruses being assembled. Again, it's unique for certain viruses to have these inclusions. So if you were well versed in looking at cells, you would be able to tell the difference between infection, not infection. Okay, so here's release. Good view of budding on all these scanning EMs. There's a transmission EM with budding. By the way, all of these are HIV. And you can see a good example of budding right there. Now, these little red dots that are on the outside of the host cell, they've been put there, coded for by the viral genome. So when the viral structure buds out of the cell and it takes the membrane with it, membrane of the cell, which becomes the envelope, it already has those little proteins studded on the outside. These are the attachment structures for the next cell it's going to take over. Alright, so those are lytic infections. Some animal viruses go into latency, which is dormancy. The herpes viridae family does this. Cytomegalovirus, varicella zoster, which causes shingles and chickenpox, Epstein-Barr virus that causes infectious mono, herpes 1 and 2, genital herpes. There's some others in the family. All right, so look at the picture over here, and you see the virus. It gets in at the lips. It moves up a nerve. Usually it's the trigeminal cranial nerve, which is nerve number 5. And right here, there is a cluster of cell bodies of the neurons. It's called a ganglion. You see it right down there also. So the virus now is in neurons, right? Because that's what's inside of nerves. You see the neurons down here as green. The virus moves into the cell body, and because this is not its normal cell to destroy, that would be skin cells, for example, inside of a neuron, the neuron doesn't do things the way the virus, to enhance the virus's ability to produce a lytic cycle. So it's not that the virus is choosing to become latent, it's the fact that the virus is now in a neuron in which it cannot come to fruition in its replication. It's kind of not quite a dead end, but its host cell is not helping it make viral structures. And so it goes into a latency. Now something can activate the virus, and it will move back down an axon to the site where the original infection was, causing a lytic infection in the cells right here, or it could be genital structures. So this is activation. Lots of chemicals, hormones, sunlight, stress, lots of things can activate. The retrovirus group with HIV can do something similar. Okay, so here's a look, a comparison of animal virus to bacteriophage. You can stay here longer if you like. I'm going to go on. All right, so what does the virus do? Well, here, staying on the idea of herpes virus, here's a look at herpes lesions inside the mouth. So you can see damage to the host cell tissues. That's called cytopathic effect. Basically, it's a lytic infection resulting in cell injury. In addition, when cells are damaged, taken over by a virus, they send out a chemical SOS, a chemical alert. And that's what's going on right down here. So the virus has gotten into a host cell. Once a virus gets into the host cell, there's no saving the host cell at that point in time. So this virus sends out a chemical SOS right here, interferons. They then diffuse into surrounding cells. The surrounding cells now will produce defensive proteins. And those defensive proteins will block these viruses from getting into them. So the interferons are a chemical SOS that then directs uninfected cells to make their own viral defense proteins called interferon, because it interferes with the viral replication in the uninfected cells when the virus tries to take them over. Latency, I've already talked about. All right, then change into cancer cells. We'll talk more about that on the next shot. Okay, lots of viruses play a role in cancer. It's estimated maybe 20-25% of all cancers are viral related. There are a lot of chemicals and hormones that can be viral related. There are genetic causes of cancers and so on, but we're talking about viruses. And viruses that can do that are called oncogenic viruses, obviously. What they do is they turn on genes in your cells that result in your cells dividing, uncontrolled mitotic division. Those genes that get turned on are called oncogenes. So you see that in the picture right here. Inside of the cell, right there, you see the genetic information of the cell. You see something called a proto-oncogene. In fact, you see it right here also. Here is the introduction of the virus. The virus injects some of its own genetic information, and this viral gene now somehow activates an oncogene which has been turned off, proto-oncogene, not read. That oncogene now, being read, causes the cell, look right here, to divide. So it causes out-of-control cell division. The view you're looking at over here at the left is HPV, human papillomavirus, and genital warts on a penis. Genital warts are benign cancers. So here's some of the things we talked about, going from left to right. Lytic infections, acute infections, fast turnaround. Infection, fast symptoms and injury, and fast death of cells or destruction of cells. Not necessarily the death of the human, though. So we have rabies and influenza and lots of others. Cold viruses would be another example. Then, to the right of that, the herpes group, latency. The virus goes underground. Then, to the right of that, chronic infections, very slow release of the virus. Remember that budding and release of the virus can occur very, very slowly. Then, far right, the conversion of a normal cell into cancer cells. Hepatitis B, Epstein-Barr virus, HPV, related, of course, to cervical cancer, EBV related to nasopharyngeal cancers and Burkitt's lymphoma, hepatitis B related to liver cancers. All right, so if you want to raise viruses, produce them in a lab, how do you do it? Well, why would you want to do that? One, you could be making vaccines. You need virus and virus structures. Two, you could be doing research on viruses. Obviously, there are reasons why you might want to have viruses. You could be a bioterrorist for that matter, too. Anyway, there are a couple of different ways. You can produce viruses in whole organisms. So, for example, in our lab, we are going to use bacterial viruses, and we're going to use E. coli bacteria as the host for the virus. And that's what you see right over there. Here's a dish of E. coli growing. And notice the little, they look like bubbles in the media, but they're not. They're little open areas of dead bacterial cells. They're called plaques. So you can actually see the destruction of the bacterial cells in the media. You could use whole plants and whole animals. For example, we could infect a mouse or many hundreds of mice with our virus. We can use embryonated eggs. This is how flu viruses are grown for vaccines. They are grown inside the yolk sac of an embryonated egg. Or you can use cells, not whole organisms, but cells of an organism to grow viruses. And that's called tissue culture. That's what you see right here. So you're looking at cells that have been infected with a virus. Now there's some normal cells right here. When uninfected, they grow very nice, uniform. They touch each other. They grow as a single layer on the bottom of the container, which you see right over here. They'd grow down here at the bottom. But look at some of these other cells. These guys over here, these guys, they're pulling away off of the bottom of the container. They're rounding up. They look like something is wrong, and that's, again, cytopathic effect. Again, here's another look at how we are going to use bacteriophages, the making of that plate up at the top right. Okay, so to finish up with all this chapter, I mentioned that there are things smaller than viruses. Well, here we are, the viroids. Look at the rose plant that you see at the top right. This is a mosaic virus of rose bushes. Is causing the destruction of the leaves. I mentioned earlier that tulips get viroid infections, which causes a variety of color changes and patterns on their petals. These are not important from a human medical standpoint, but they would be important if you're a rose grower or in the case of a tulip grower, because you can sell your bulbs for many times the amount. If they've got these beautiful patterns, people will pay a lot more for the bulbs. Anyway, viroids are only plant diseases, and they're only RNA. Only RNA. Now, let's go to the prions. A prion, your cells have got normal prion. It's called prion protein. It's on the outside of the cell membrane, and it is a folded protein associated with neurons, and it has to do with transport of material in and out of the cell. There are infectious prions that you can ingest, and these infectious prions mess up your own neuron prions, causing a malformation of the folding pattern. This destroys the neuron. It can't function properly. So what you're looking down at the bottom here is brain tissue. And this is full of neurons. And look at these large holes in the brain tissue. They are not supposed to be there. This is destruction of brain tissue due to the problem of the malfolded proteins of the neurons created by infectious prions. The word prion means it refers to infectious protein. These diseases that are mediated by prions, they're called prion diseases, are called spongiform encephalopathies. Spongiform because this is what happens to your brain. And the symptoms are all very similar for these prion diseases. They're called encephalopathies because they destroy the brain. It's kind of like a senile dementia. If you have a grandparent, great-grandparent that's 80, 90, 100, and you know what dementia is. Well, that's what happens in all of these encephalopathies. So here's a look at some prion diseases in humans. There are quite a few of them in other animals also. But the main one is Creutzfeldt-Jakob disease, CJD. I mentioned that they all have similar symptomology. They're very, very slow incubation periods. Look over here under incubation, months to years. By the time you have the symptoms, it can be a very long time after you were infected. So there's really no way to determine if you have been infected until you get the symptoms. Notice when you're looking at the symptoms over here, they're all progressive dementias. And progressive, they get worse, 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 worse. Different kinds of prions infect different parts of the brain. Here's what is called mad cow disease or bovine spongiform encephalopathy. Here is location of destruction of CJD disease. And this is a look at a adolescent boy with a kuru that was first identified in New Guinea, one of the New Guinea islands. We've talked about antibiotics and synthetic agents already, and I think I mentioned some antivirals, but here's another look at antivirals. The bacterial antibiotics and the drugs used for fungi and protozoa are not functional at all against viruses. Remember, there is no similar target for viruses that you have for bacteria and fungi and so on. So you have to totally come at this with a different approach. So you see a life cycle right here in the middle of the page. And here is your attachment, and there is your entry of the virus, and here is your synthesis stage, viral proteins and DNA. Here is your assembly, and here is your release. And at every one of these stages, you'll notice that there's some drugs listed which block that particular stage. Again, here's a chart showing the same kind of thing. So, up at the top, you've got fusion inhibitors. They block the attachment of the virus. Interferons, I've already mentioned. They will cause an uninfected cell to make its own defensive proteins that block viral infection. Then there are reverse transcriptase inhibitors for HIV, because that blocks the production of DNA from RNA. And there are a lot of drugs that act as nucleotide analogs, like a drug that looks like uracil, or a drug that looks like adenine. And so the host cell picks up the drug, thinking that it's adenine or uracil, when it's trying to construct viral genetic information, and it can't do it. Up here at the top, you see neuraminase inhibitors like Tamiflu that work in blocking the attachment and the release of flu viruses from infected cells. And again, you can stop and look at this in more detail. And again, here's another shot from your book comparing bacteria to viruses to viroids to prions. Take a look at that. Be sure you understand these four different groups: the viruses, viroids, prions. And just a shot at some viral diseases. At the bottom is an iron lung machine that was used for polio patients that had paralysis because the poliovirus destroys motor neurons in the spinal cord. And over on the right is from the 1918 flu pandemic, that infected a vast amount of the world. Notice the filter pad that they're wearing over their mouth and nose. And that's it.